and speak here at the second Thailand Hub of Talents in Cancer Immunotherapy International Conference. I'm sorry that I can't be there today, but it looks like a really fantastic meeting. And so I'm going to tell you a little bit about the work that my lab does, trying to understand how tumors interact with the immune system during metastasis. And so I'll start by highlighting the fact that if you look across the range of solid organ malignancies, these cancers will exhibit differences in their distant metastatic proclivities. They'll spread to different sites depending on the different malignancies. But what they share in common is that all of these cancers really spread to lymph nodes, typically local regional lymph nodes, but also distant lymph nodes. And often this occurs prior to dissemination to distant tissues and really portends worse prognosis for these patients. So I'm showing you melanoma here on the top, where you can appreciate that patients who have positive sentinel lymph nodes do much worse than patients that don't. And this extends really across a wide range of solid organ malignancies. I'm showing you breast and pancreatic here, where not only does the number of nodes spell worse prognosis, but the ratio of involved to uninvolved lymph nodes portends worse prognosis for these cancers. And so to date, there have really been two predominant models for how cancer spreads. And the first was posited by a turn of the 20th century surgeon, William Halsted. And what he suggests is that tumors spread first through afferent lymphatics and reach lymph nodes. And then once they do, they might acquire some additional metastatic traits that endow them with the capacity to then leave through efferent lymphatics and spread to distant tissues. But now, with the advent of various sequencing technologies, what researchers have been trying to do is reconstruct phylogenetic relationships between tumors and distant sites and primary tumors. And what they've begun to suggest is that perhaps what spreads to lymph nodes is not necessarily what spreads to these distant tissues. Perhaps these things spread in parallel. Maybe we see them first in lymph nodes because of lymphatic drainage, but the tumors that go there aren't necessarily what seeds the distant metastases, and these lymph node metastases play no functional role in further tumor progression. But my lab is an immunology lab, and as immunologists, we recognize that lymph nodes should harbor the vast majority of potentially tumor-reactive lymphocytes. So one might expect that if a tumor does get to a lymph node, it should encounter those lymphocytes, arrest, and ultimately be eliminated. And yet we know frequently that's not the case, and these patients will go on to develop distant metastases. One other thing I want to draw your attention to is when we look in the context of immunotherapy, we can really begin to understand how important systemic immune responses are. So this was a study that we published quite a few years back now, and so I won't go into it in any major detail.
Recording in progress.
In this mouse model is a breast cancer mouse model where PD-1 is ineffective, so we use anti-PD-1 on this context, or we use an effective immunotherapy.
เครื่องไหนเอ่ย
สรุปปิดมายังพี่
ปิดยัง
across their bodies and subject them to immune profiling by mass cytometry. Now, I'm not going to go into details of these analyses.
Okay.
Tumor microenvironment. What we're doing here is we're following changes in these various immune populations, either increase of immunotherapy or really they decrease anti-immunotherapy. What you see is in the tumor microenvironment, almost nothing is changing when you have effective versus ineffective immunotherapy. Definitely there are changes in immune populations, but nothing really distinguishing these immune responses to effective immunotherapy. When you look at a tissue like the lymph nodes, it's quite different. So in lymph nodes, you can see that there are a whole host of B and T cells that are activated and proliferating when you talk about effective immune responses, but they're not going to ineffective immune responses. So this work really demonstrated that it's not so much about just reinvigorating immune cells within a local tumor microenvironment, but effective immune responses really do require systemic administration of anti-tumor immunity inside And so the first thing we really wanted to have was the presence of these lymph node metastases, the something to promote metastatic seeding or colonization of different tissues. So we asked the question, we use a fairly simple experiment, but I'll go through it slowly because it is rather important for the studies I'm going to talk about today. So what we'll do is we'll take mice and we'll put in either this non-metastatic parental cell or one of these lymph node metastatic these mice. We'll wait a couple of weeks for it to condition the mouse, and then we'll challenge this mouse with a tailgating injection of the fluorescent version of the parental cells. And so for those of you who aren't familiar with these assays, when you inject cells into the lateral pelvis in mice, the first papillary bed that they encounter is the lungs. So this is a way of proving how good a given population is at seeding lung metastases. And so we're asking what presence of these lymph node mets does something to condition the host to render it more susceptible to lung colonization by these tumor cells than does parental cells. And that's exactly what we see. So mice starting these lymph node metastatic cells had more lung mets when challenged with these tailgate injections than did mice carrying parental lung metastatic cells. And so we think that lymph node metastases are doing something to promote seeding different metastases. So what are we going on? So we have all of these different cell lines. So the first thing we wanted to ask was different about this. So we subjected them to bulk RNA sequencing. You can see there's a whole host of differentially expressed genes. And if you look at what these genes are and perform your favorite types of analyses, whether that's flow analyses or gene set enrichment analysis, what really pops up almost every time is that many of these genes are not only related to the immune system, but in fact related to both type 1 and type 2 interferon responses. So these interferon-stimulated genes are frequently upregulated in the context of lymph node metastasis, at least within the context of a cell line. We also wanted to make sure that this is something that's not specific to our esoteric model system. And so we went back and tried to analyze patients here at Stanford. So we took patients with head and neck cancer. We sorted out their malignant cells and subjected those to RNA sequencing. You compare it to our mouse lines. And so what I'm showing you on the y-axis here is differential gene expression in human head and neck cancer patients comparing patients who did or did not have lymph node metastases. And on the x-axis, what I'm showing you is our mouse data, differential gene expression of our lymph node metastatic cell lines, so upregulated context of lymph node metastasis. And what you see is if you look at what's conserved and upregulated in both these data sets, again, you're looking at different tumors and different species, but the vast majority of these genes, 24 and 27 genes, are related to interferon responses. And so we think this upregulation of interferon-stimulated genes, ISGs, is something that's conserved across lymph node metastasis in a range of different cancer diseases. So one thing to be noticed is that if you took these cell lines out in culture, you could take them many times. They maintained expression of these ISGs, even in the absence of exogenous interference. So the thing you want to know is whether or not there's something to do with genetic wiring that may be instilling these phenotypes in these lymph node metastases. And so we performed TAC-seq, which is the way of looking at changes in chromatin accessibility across the genome. What you see in green is that if you look at these third and seventh generation lymph node lines, you have increased accessibility in transcription start sites of various IFGs highlighted in green, but also at cis-regulatory regions in blue, the sort of enhancer regions upstream and downstream of the genes of interest, or these ISGs. Furthermore, we can perform transcription factor motif analysis. We'll see that not only do we get enrichment in SOS family transcription factors, which have been well associated with metastasis, but we also see enrichment or increased activity of a variety of STAT family transcription factors, which are key regulators of interferon signaling. And so are these things important? And we think they are. We can take our lymph node metastatic cells and knock out a variety of these transcription factors. I'm showing you the SMART1 knockout right here. And when we do that, you see a reduction in lymph node metastases of these lymph node metastatic cells. So it seems like this constitutive ISG signature is really critical for promoting metastasis. Of ISG, hundreds, maybe thousands of these genes, but there are two that stand out in particular class as immunologists, and those are PD-L1 and MHC class 1, which you can see in both of these genes at the transcriptional level are gradually upregulated with increase in lymph node metastatic potential. So CD274, which is the gene that encodes PD-L1, is gradually upregulated, going from the parental over here on the left all the way to these ninth generation cell lines on the right. And you see the same thing is the case with the beta 2 microglobulin, which is the common subunit of MHC class 1 molecules. We see the different proteins as well. So in red, you can see a variety of these LN6 lines, all of which have upregulation of PD-L1 and MHC class 1. And I'd also draw your attention to, and that's at the third level on the surface level, but I'd also draw your attention to, in blue, you see P16F10, which is something that's been selected from metastasis to the lung. You can see that in this case, there is no downregulation of PD-L1 and MHC class 1. Suggesting that these phenotypes are really unknown metastasis specific. So why be upregulating the genes, particularly once you get upregulating the C class 1 when that should make the tumor more susceptible to killing by CD8 T cells? And we thought this has something to do with evasion of natural killer cells. Natural killer cells have receptors that MHC class 1. So we thought lots of tumors are upregulating MHC class 1 to evade NK cells. So we took our tumor cells and we cultured them with NK cells and looked at killing. And what you see is that indeed the lymph node metastatic cells are more resistant to NK cell mediated cytotoxicity than is the parental. And we can go and deplete NK cells in mice that we implant the parental non-metastatic to. And you can see that when we get rid of NK cells, we have an increase in lymph node metastases. And so in the inverse, we can take our lymph node metastatic cells, knock out MHC class 1, and you see that when we do that, we get a reduction in lymph node metastases. But if we then come in and deplete NK cells, we can rescue the ability to get MHC class 1 knocked out to metastasize. So we think that the regulation of MHC class 1 is the mechanism by which these tumors are evading NK cells and metastasizing their lymph nodes. PD-L1, similarly, as you might expect, we believe has something to do with evasion of T cell responses. And so we can come in, you see that if we knock out PD-L1, we get a reduction in lymph node metastases of our lymph node metastatic arm. And we can overexpress it in our parental lines, and when we do that, we get an increase in lymph node metastases. And then finally, just what I showed you with the NK cells, if we deplete, if we knock PD-L1, we get a reduction in lymph node metastases. And I just showed you, but if we then go in and deplete T cells, we can rescue that knockout line's ability to metastasize the lymph nodes. So we think that the upregulation of PD-L1 is a mechanism by which these lymph node metastases suppress T cell responses and ultimately colonize these lymph nodes. So to take a step back, what I've shown you so far is that you think we have chronic tumors. They initially upregulate MHC class 1 that confers the ability of those tumors to evade NK cell responses and get to lymph nodes. Well, if they get there, there's chronic interferon, and I didn't show you much of these data today, but so I could say that the chronic exposure to type 1 or type 2 interferons resulting in this epigenetic rewiring that's leaving these various ISG loci open, such that even in the absence of exogenous interferon, these tumors maintain expression of these various ISGs, chief among them being things not only in the acute form, but PD-L1, which allows these tumors to suppress local T cell responses and ultimately colonize these cells. So then we will talk, I want to come back to this concept of how does the presence of lymph node metastases promote metastasis to lymph tissues. So I remind you of this experiment that I showed you before, where we'll put in either these parental non-metastatic tumors or these lymph node metastatic tumors in the mice, challenge them with tail-vein injections. And what we showed you is that mice-bearing these lymph node metastatic tumors had more lung metastases than mice-bearing renal tumors. Again, suggesting that the lymph node mets are doing something to condition the host to render it more susceptible to metastatic colonization of different tissues. So our initial study on understanding how tumors affect lymph nodes really focused on understanding immune responses in their role in promoting this response. So we thought that the metastasis of different tissues are also related to immune responses. So to ask these questions, what we did is we went back and repeated these studies, putting in the parental or lymph node metastatic tumors and challenging these mice with tail-vein injections of the parental. But instead of doing this in we did it in NSG mice, or mice that lack they're really your whole lymph sample, your B cells, T cells, T cells. So we're asking whether or not you still get this enhanced seeding of lung metastases. And the answer is that you don't. So mice bearing the lymph node metastatic tumors have the same number of lung mets as those bearing legal tumor, suggesting that whatever effects the lymph node mets are having on promoting metastatic seeding of distant tissues is happening at least in part through the same point. So the next question we wanted to ask was whether or not these effects are tumor specific. So we go back and we repeat these same types of experiments, now going back into wild-type mice, wholly immunocompetent mice, but instead of challenging the mice with a tail vein injection of the parental tumors, we instead challenge the mice with a tail vein injection of an unrelated tumor. So this was MC38 colorectal cancer, still syngeneic in B6 mice, but unrelated to this parental lymphatic tumor that we put into the mice. What we see is that when we repeat these studies, there's no difference in the attack. So whatever the lymph node mets are having to promote lung metastasis, it's happening in a tumor-specific manner. And the last question we wanted to ask is whether the lymph nodes themselves were really critical for the promotion of these different mets, or whether it was just a phenotype that was required during metastasis. And so we go back, we put the lymph node metastatic tumors in the mice, challenge the mice with tail vein injections again, except in this case, we're doing it in mice that lack lymph nodes. So these are LT-alpha knockout mice. And what you see is that these mice that don't have lymph nodes, you have a reduction in lung metastases, suggesting that whatever's happening in the lymph nodes and the sort of architecture therein is really critical for the promotion of distant. The next thing we want to know is whether or not the cells within those lymph nodes are really active for this effect. So what we do is we do an adoptive transfer study. Now I don't want to go through this in a whole lot of detail, but suffice it to say that we take mice, all of them have these primary tumors, including parental tumor cell, and then half the mice will go in and do a surgery and put the lymph node metastatic tumors directly into lymph nodes, and half of them get a sham injection. We do this mainly to control time. And then we come 4 days later and we take the lymph nodes out of these mice and fast purify all the immune cells and transfer those immune cells now to new mice that we then come in and challenge with tail vein injections. And whether or not there's something about the cells and the lymph nodes of mice with lymph node met that promotes metastatic seeding. And indeed, that's the case. So mice that receive leukocytes from mice with lymph node mets had more lung mets than those that received a big number of immune cells with the mice without metastasis. And so what's going on in these lymph nodes? Well, as anyone should do these days, we went in and performed a bit of single-cell RNA sequencing. And so we looked across a range of various immune populations within lymph nodes in the context of mice that had no tumors or parental tumors only, or lymph node metastatic tumors. So what you can see is that there's a whole range of changes that happen in the B cells, and the T cell shifts away from naive populations toward more activated populations, many of which have a real IHG signature as well. And within the normal populations, we see similar types of shifts as you might expect. So mice bearing lymph node metastatic tumors, and these lymph node metastases had more macrophages. They had, these tend to be immunosuppressed macrophages. They had more neutrophils that are consistent with immunosuppressive phenotype. And they also have reduced numbers of these cross-presenting CDC1 dendritic cells, so these XCR1 positive dendritic cells. All of it makes sense for the generation of sort of a more immune tolerant state for lymph node organization. What I want to draw your attention to is that mice bearing these lymph node metastatic tumors had more Tregs within their lymph nodes than mice that had the colorectal tumor. So furthermore, if you take the CD4 T cells and co-culture them with the lymph node metastatic tumors, they result in more induction or more differentiation into Tregs than if you co-culture these naive cells with the parental tumors. And this is functionally important because we can go in and deplete Tregs in mice that have these lymph node metastatic tumors, challenging with tail vein injections just like before. And if we get rid of Tregs, these are FOXP3 hormones that we can deplete Tregs with by administering diphenotoxin. If we get rid of these Tregs, you see we have a stark reduction in lung. So with Tregs, we're certainly lung metastasis. We also see this is the case in humans. And so these are head and neck cancer patients, and you can see that those that have lymph node metastases have more Tregs in their lymph nodes than those patients that didn't. So just because we know that deficient Tregs results in fewer lung metastases, you might be thinking, wait, perhaps we know that Tregs promote cancer, and so is there anything specific about the Tregs within the lymph nodes? And so to start to ask these questions, we do a series of studies, and I'll show you kind of a simplified version of what we did here, although you can read the full version in the manuscript if you're interested. But essentially what we do is we take mice, all of which have primary tumors, and half of which have lymph node metastases and half of which don't. And these are FOXP3 GFP mice, so what we'll do is we'll take out the Treg, we'll sort them out, and transfer them to new mice that will be challenged with tail vein injections. So we're asking if there's anything specific about the Tregs and the lymph nodes of mice with lymph node mets that's different from the Tregs in mice without lymph node mets. And that's exactly what we see. So mice that receive Tregs, the same number of Tregs, from mice with lymph node metastases, have more lung mets upon tailgate challenge for tumor cells than the mice that receive Tregs from lymph nodes of mice that just had tumors. So there's something specific about those lymph nodes, those Tregs in those lymph nodes. You may be wondering if they're antigen specific. So to ask those questions, we went in and performed Reticker-Flynn analyses. So we took mice, all of which had primary tumors that expressed OVA, and half of which had lymph node mets that also expressed OVA, and half of them had no lymph node mets. We went in and performed pain, which you can see that the mice that the Tregs and the lymph nodes, the mice with lymph node mets, over-specific Tregs as compared to the Tregs in mice that just had primary tumors that express OVA. And this is functionally important. So we can go in and do studies wherein we put in the parental or lymph node metastatic tumors into mice. And these are not OVA expressing tumors, but we'll put them into OT2 mice where the CD4 compartment and the tumor have TCRs that specifically recognize OVA. And so we're asking the question, these mice can no longer, for Tregs, can no longer recognize the tumor agents. Do they still promote metastatic seeding of different sites? And the answer is that they don't. So there was no difference in the lymph node carcinoma on tailgate challenge in these mice with the lymph node metastatic tumors as compared to parental in these OT2 mice, suggesting that really the recognition of tumor antigens by the CD4 compartment and presumably by the Tregs is really critical for the promotion of this inhibitor. And so the summary I've shown you so far, you have primary tumors, which will everything MHC possible, and that confers the ability for them to evade NK cells and get to lymph nodes. Whereupon they're exposed to chronic interferon signaling in a manner that results in epigenetic rewiring and constitutive expression of various IMGs, chief among them being things like PD-L1, which allows them to locally suppress T-cell responses and colonize T-cells. And then once they do, they activate the naive CD4 T-cell compartment to induce antigen-specific tumor-specific Tregs, and then presumably recirculate throughout the host, leaving with them tumors in a manner that later these distant tissues amenable to metastatic colonization. And so in contrast to the prevailing models, the Alpenian model where tumors from first the lymph nodes, or leave lymph nodes and spread to distant tissues, or the systemic model where everything spreads in parallel, what we've been suggesting is a new model that we like to call metastatic tolerance. In our model, tumors spread from the lymph nodes, whereupon they use this tumor-specific immune tolerance that becomes systemic, and in doing so renders these distant tissues amenable to metastatic colonization. Tumor or perhaps from a lymph node metastasis. So that antecedent nature of lymph node colonization is critical, but not because it's serving necessarily as a cellular reservoir of distant metastases, but rather because it establishes the systemic tumor-specific immune tolerance that renders these distant tissues amenable to metastatic colonization. So I think I just want to close by highlighting the fact that I think in the context of immunotherapy and also in the context of metastatic progression, lymph nodes play these really critical roles. They can be really important for driving anti-tumor immune responses, in really orchestrating systemic anti-tumor immunity. They're actually promoting metastasis in the context of induction of systemic immune tolerance. And so if you think about going forward with how we should treat cancer patients, particularly how we should treat metastatic disease, I think we have to be careful to not go too far in either direction. Removing all these lymph nodes could be really problematic because we then don't have site to educate anti-tumor responses. And similarly, we have to be creative about thinking how to treat these lymph node metastases in the first place. So we have highlighting some of the other stuff in my lab, probably well known. You have a range of different projects going on. A lot of them focus on understanding how tolerance is induced within lymph nodes and using a variety of tools to explore how the tolerance becomes systemic. We also have a lot of interest in understanding tumor immune co-evolutionary processes that go on during metastatic progression, particularly on the adaptive immune side. And then finally, we try to leverage the insights that we gain from these 50 areas of the lab to build new classes of immunotherapies that do things like go to lymph nodes and wave. So with that, I want to thank the various funding sources and especially all of the folks in our lab. We've been running for a little over a year, I think.
So a lot of beginning projects going on, and certainly a lot of folks that I've seen don't take a lot of risk to me. So I want to thank Ron in particular, and certainly the organizers of this meeting for the opportunity to find out today. On behalf of them, not to take any questions.
So I think Dr. Nathan is on the Zoom with us, right? So I think if there is any question, so we can ask him lively or discuss with him. ผมจะได้เห็นหน้าเขาไหมไม่ใช่ไวไม่ต้องเป็นไว
Hello, Professor Nathan. Can you hear me?
Yes, I can hear you.
Okay. Perfect.
No, this is can you be speaking the person who reached out to you in the first place. I'm great to finally hear you talk. Eventually, it was a great talk. Thank you for accepting our invitation and talking to us today. My question is, could you please go back and explain about the response of the cells in the lymph node when they are stimulated by interferon, and how does that influence the epigenetic reprogramming? Because my thought is that interferon exposure would theoretically suppress those cells and arrest those cells. How does the constant beating of those cells with interferons induce the tolerogenic genotype? Thank you.
Yeah, absolutely. Well, first of all, thanks so much for the invitation, and I'll echo Richard's sentiments. I'm sorry that I'm not there in person, but yeah, actually, that's a really good question. It's something that we think about a lot, and I think what happens is there's probably a lot of parallels to what we see in lymph node metastasis with what people like Andy Min and even John Weary have been describing in the context of therapy responses. So I think that initially maybe tumors get to lymph nodes, or even before they get there, they're exposed to potent, good anti-tumor immune responses. Even if the primary tumor is growing, I think there's probably good anti-tumor immunity that involves a lot of interferon signaling. And then I think what happens is if a few of these cells manage to survive within lymph nodes, that chronic exposure, and it really has to be chronic, but that chronic exposure to interferons is what's stimulating this sort of epigenetic rewiring. And maybe it's the same kind of things that Andy and other people are describing in the context of radiation therapy or immunotherapy. But ultimately, that epigenetic rewiring results in these cells leaving these ISG loci open, such that even if there's not any exogenous interferon around, the tumor cells still maintain that signature. And so you're not getting the benefit of all the interferons acting on the T cells and immune cells, but you still get this kind of this interferon signature within the tumor cells, which results in upregulation of things like PD-L1. I don't think we've worked it out entirely how it's happening. We're trying to do a series of studies right now to get at exactly the molecular mechanisms by which this rewiring is happening and the really dependencies of the different interferon receptors in driving it. But we do tend to see this at really an epigenetic level as a consequence of really the long-term chronic exposure to interferons. I don't know if that answered your question.
Absolutely, it does. Thank you.
Can I ask another question? Nathan, thank you so much for your talk. My name is Natia. I have a question about one of your last slides. You say that you're trying to do genetic engineering to tackle these torrents. Is it involves a regulatory T cell?
Yeah, so a good chunk of our lab is interested in Tregs. I mean, unfortunately, a lot of the therapies targeting Tregs have not been super effective to date. So really the way we think about the therapeutic kind of arm of my lab is that we try to essentially engineer. We do some T cell engineering now as well. We also do some other weird stuff with bacteria and things, but most of it's focused on engineering T cells to go to lymph nodes, but really the goal there is to go to sites where there are lymph node metastases and try to reprogram the immune response away from tolerance and towards immune activation. So we use CAR-Ts and transgenic TCR T cells to go to those sites, but we're not really as focused with having a lot of lytic activity from our CARs or from our engineered T cells, we're really using them maybe kind of like Richard in it, but in a very different way, to get to these sites, these lymph nodes, and then activate the endogenous immune response against tumors. Try to switch it away from tolerance. So we're kind of like trying to figure out ways to stop Treg induction and really push or shift the balance towards activation.
Okay, I think, Professor Lee, please.
Thank you for the wonderful talk. So you showed the Treg cell from the subcutaneous lymph node tumor in promoted metastasis. So where did you isolate the Tregs? Is it from lymph node or spleen?
It's lymph node. Yeah, that's a good question. I mean, I think sometimes, if you look systemically, you often don't see like massive shifts in the Treg frequencies, but I think there's probably more going on there in terms of their specificities that we're not picking up on. So there might be hints of things in the spleen, but we haven't seen that at just like a, broad sort of immunophenotyping level. Is that what you were asking about?
Okay.
Okay.
Thank you, Professor, and Damasan from Chiang Mai University. So I just curious that the tumor originations, some tumor identify to metastasize to the lymph node, but some tumor is try to go to the intravenous system. Do you think why is a tumor they select to go in which direction, and how different of the immune response of the body to these two kinds of tumor?
Just to clarify, you're saying between things that go through the lymph to lymph nodes versus like through the blood to distant tissues?
Yes.
Yeah. So I mean, it's a great question, and one we think about quite a bit. And if you start to mess around with the immune system in these mice, you do sort of start to see different routes of metastasis, which is something we spend a lot of time thinking about. I think that there's probably an element of what gets to lymph first due to the drainage of lymphatics, and so maybe like draining there is easier, maybe surviving there is not so easy. But I do think that the phenotype that we see in these lymph node metastatic cells is one wherein they're not entirely capable of just evading immune responses. So we don't see, for example, we don't see just like a total loss of neoantigens or anything like that. And really what I think happens is they're inducing one type of immunosuppressive phenotype. And then when we look at what is able to get to the lungs or something, you often see things like loss of MHC class 1. So I think that might be a different mechanism of evading, cells in those sites. And so I think there's an element that's just about the microenvironment and the differences that these tissues present to these tumors, and certainly things like ferroptosis might be different in these different tissues. But then I also think that the immune compositions and sort of the stage they are in tumor progression really plays a big role. So maybe once you've gotten to a lymph node, you've induced these Tregs, you've overcome one barrier such that then a different phenotype of cell could make it to the lungs or the liver or something else like that. It's just a hypothesis, though. I don't have strong data to suggest that. Is that what you were asking about?
Thank you.